that although my role is Chief Medical Officer and responsible for clinical development, I've noticed how relevant is becoming to my role to ensure that I understand all regulatory aspects that are involved in gene therapy. And they're changing extraordinarily quickly, especially in the area that I'm going to be discussed today, which is where we're focused in hemophilia A. So in this first slide, I have an overview, an agenda of my presentation today. I'm going to discuss the key FDA regulations for gene therapy, focus on safety, and I'm going to give you some sense about the way we interact with the agency here in the United States, and also our expedited programs, how we're able to adjust them to what the agency is offering to us. I must also say that although I'm not going to discuss specifically details from the European regulatory agency, or even others in other countries, I think that everyone is really benchmarking each other. So everything that I'm going to discuss here, it really applies to the regulatory needs somewhere else. I'm going to also discuss an area that, again, as a chief medical officer, you're not supposed to be very involved. When I was in my career now of over 30 years, excuse me, in the industry, I really never thought that I should be involved in the manufacturing of the drug product that I'm going to use in my clinical trials and later on for commercialization. But this is becoming extraordinarily important in this space, in the space of gene therapy, and by the way, also in cell therapies. And I'm going to use now a case study, which is the work we are doing with factor VIII replacement, gene replacement therapy, and for hemophilia A, as one way to explain you how we are doing all these aspects, how are we interacting with the agencies, and how we are also doing all this development. So again, with all of that, I hope I will give you a sense about my daily work in some way. Starting then with the key FDA regulations, the regulatory pathway for any type of drug development is always the same. You start with your phase 1 investigational studies. The objective is to design your right type of assessment of the patients in terms of safety and efficacy. Then you have your phase 2, where you look in this case for the type dose, the right dose. So this is a dose escalation type of approach. And then you have the pivotal trials, phase 3, and eventually a phase 4. But what happens with gene therapy is that the agencies are already recognizing that we can combine phase 1 and phase 2. It's more phase 2A. That means that we're looking into safety and a dose ranging type of assessment that will give us a sense about the right dose for the patient. So we're combining safety and dose ranging studies. And I think that that's something that is also very relevant in this space. There is also something that is very interesting in these kind of studies. Since these are monogenic diseases, these are diseases that really happen only in certain individuals. You cannot really have a controlled arm. So even for the pivotal trial, our studies are are just one single arm. Are single arm type of studies, so they are not randomized control studies. Now, when interacting with any kind of agency, there are a number of conversations that really need to happen. And in this type of conversations, in the US, we call it Pre-IND. This is a meeting that sometimes you have even a pre-IND that the agency calls it interact. And this meeting tries to ensure that you achieve the clearance at some point when you do a submission. We're going to see that in a moment, for the investigational new drug, the IND. And the key focus of the agencies is always safety. So they want to make sure that whatever type of, and especially when you do a gene transfer, and you're practically looking into a very concrete type of approach to patients with something that has not been tried before, that the main point that you want to control is safety. So that's why it's extraordinarily important for the agencies that the necessary studies are designed to demonstrate safety in patients enrolled in these phase 1-2 studies. Additionally, very important for them is that you clarify and discuss with them exactly what type of safety and efficacy endpoints are you going to use. And you want to gain FDA support for this entire approach that you are taking. You also provide the opportunity to discuss with the agency your translational science. What does it mean? That means that you take all the information that you've gained in your point of your initial studies on cell cultures, on animal models. In our cases, for instance, they are wild type animal models, models with humanized mice, liver, and also non-human primates or monkeys. And you want to ensure that the agency understands the process of taking all this information and translating that into the doses that you're going to use in your trials. So this is translational science, and this is an opportunity. Obviously, you discuss with them several other aspects that we're going to see in a moment, related to indeed all this preclinical work, and also to the manufacturing of or in this case, of your gene transfer product. And all this establishes a platform for future interactions with the FDA. So you are now ensuring that you are able to move on with all the next steps, which are obviously the pivotal trials, and this is the basis of this interaction. Now, I'm going to very briefly discuss something that is an advantage for us working in rare disorders. And this is the fact that the FDA and also their agencies provide an opportunity to have a number of designations that are going to accelerate the review of your materials, and also they're going to accelerate the timing that you are provided for achieving an approval, and that's the orphan drug designation, fast track designation, breakthrough therapy designation, regenerative medicine advanced therapy for when you are in that space, or accelerated approval. And where I'm going to focus, which is, I think, the one that almost everyone in this space, in the rare disease business, is interested, is in the FDA orphan drug designation. We obtained that already in 2 of our programs, and we are right now filing for an orphan drug designation in Europe. And there is no orphan drug designation in China, but there is another way to do that. So again, you need to understand how the different regulatory agencies do that. And this is, there are already very clear definitions. In this case, for instance, it has to be a disease that affects to less than 200,000 patients in the US. So now let's move into key chemistry, manufacturing, and control, the so-called CMC for gene therapy. And I mentioned a moment ago that although this is not part of the role of a medical team, of the chief medical officer, this is something that is extraordinarily relevant, because you are going to be utilizing a material that is produced in a certain factory, in a center that is highly specialized for developing these gene treatments, and then it's going to be shipped to the clinical sites. And even that shipment, ensuring that everything goes well from one place to another, to goes to the pharmacy in the hospital that is preparing the actual infusion that is going to be put in the patients. Believe it or not, I am involved in that. And here is a characterization of what has happened in our industry, how we are moving into higher and higher manufacturing and characterization complexity of our drugs. And when we started with small molecules or with large molecules, that was obviously relatively practically chemistry. And then we started moving in large biologics, and I worked for almost 15 years with the AbbVie team, and also with the Humira biologic type of anti-TNF, immunoregulatory drug, and it was. We also have bioreactors. We needed to use cells to produce these monoclonal antibodies, but the type of complexity that we are reaching now with gene therapy is extraordinary. So we can compare from a jet fighter, which is what you do with biologics, to practically an international space station. And it's not only the complexity, and we're going to see a few graphs about this, of really putting this machinery together. It's also all the analytics associated to that. It's extraordinarily complex, and sometimes that's what delays really the production. The fact that you have to analyze different types of elements that are incorporated into this so that you reach a high level of quality. And when a patient receives this treatment, is really receiving the right dose and the right, what we call vector genomes, so the right amount of material that you expect. And that's not easy. It's not easy to really be able to characterize this drug and ensure that you provide what you think you are providing to the patients. So again, this is for me a learning space right now. We recently brought in one of the best industry veterans in this space. He had been working with Unicure also on hemophilia B gene therapy, and now he's with us. So he's also pointing to us a number of challenges that we need to take into account. But if we put together now the CMC, all these manufacturing aspects with the regulatory world, we see that there are a number of regulatory challenges. It's a lengthy and complicated manufacturing process that needs to be explained to the agency. You reach relatively low yields. That means that you have to do a lot of production to really get a tiny quantity of material for your patients. You need to ensure that you release the test, the batches that you're using with your patients with the right analytical procedures. And this means that it's very difficult also to move from different types of what we call bioreactors, from 500 liters to 2,000 liters to even 20,000 liters. So there are different type of aspects that are very important in manufacturing. And again, you need to be aware of. The impact of all this in your regulatory submissions is that although you need to show one single batch for the agency to be able to approve your drug, you also need to ensure that you have enough material for your clinical trial. And when you start with a relatively small clinical trial, like we are in this case with a phase 1/2A, that's just in our case up to 12 patients, but you have to start thinking about 50 or 100 patients for your pivotal trials. So you need backup plans. You need a lot of interactions with your regulatory team and with your manufacturing team to ensure that everyone works together. And this is what makes my work really interesting, because it's not just doing my standard clinical development that I've been doing over the past 30 years. It's also learning something new and working as a team. Now, I'm going to spend a moment describing some aspects of our hemophilia A gene therapy program. And here, let me start with hemophilia. Hemophilia is a monogenic disease affecting exclusively the X chromosome, and as such, you are expected to have this disease, and it happens indeed in the majority of the patients are males. Some women still, because they are carriers, have some bleeding disorders, but that's the majority. One aspect that I'm highlighting here is the fact that because these patients receive replacement factor VIII during sometimes the majority of their lives, they develop inhibitory antibodies. And this makes it extremely important to ensure that you look into those patients and consider if they really need or they can receive gene therapy if indeed they are going to inhibit the factor VIII that they're receiving. So that's a possibility, and we're working on that as an industry. But again, we think that we are already opportunities very well. Now, here is a busy slide, and let me tell you why I have first these two pictures to the left and to the right with rocket, and also with module, a lunar module, and a capsule. And this is because I'm going to use as a reference the Apollo 11 journey to the moon, to give you a sense about what do we do in our case. First, we have the AAV, which is the vector. In our case, we use the AAV8, which includes a specific promoter and a factor VIII transgene that in our case is unique. And let me explain you a little bit why this is relevant. The fact that we have a transgene that has 90% of human sequences and 9% of porcine sequences. It is important because although the field of hemophilia A gene assessment and analysis has been going on for several decades, and we have already over about 2,000 patients probably treated in different ways with gene therapy for factor VIII, we having always been using a fully human, 100% human sequence. We have been working with the Emory University, which has been working on ancestral sequences for factor VIII, and they identified these 9% of sequences that were able to increase the secretion of and biosynthesis of factor VIII. So that's what differentiates the way we're doing our job at ASC Therapeutics. So we have an engineered liver-specific promoter that minimizes the size of the payload that is going to be incorporated into the rocket, and this promoter is the capsule that you see here in the Apollo 11, and then we would have the engineered liver-specific factor VIII transgene, which is the actual lunar module, and we're going to see how it works when it starts approaching to the moon. And we are going to reach, because of this combination of promoter and liver-specific factor VIII chimeric transgene, a higher secretion and expression of factor VIII in the liver. So here is another graph depicting, first to the left, you have that what we do is a one and done. It's one time. We don't have any opportunity to redose these patients. And what happens is that once we have our construct, which includes again the promoter and the transgene into the rocket or the AAV8 in our case, it incorporates into the cytoplasm. From the cytoplasm moves into the nucleus, where it releases the payload, which is the promoter and the actual transgene. But this transgene is circling the chromosomes. So it's like the lunar module still not landing in the moon. It's around the moon, is just ensuring that even being outside, as we call as episome, able to deliver the factor VIII through all the machinery in the cell. And what happens is that here we have first the transcription, so that the DNA is transcribed into mRNA. The mRNA is then traduced into a protein. And what happens here is there is a folding. So we give the instructions for the factor VIII to be folded. And in this folding is where the transgene that we are using with chimeric sequences from factor VIII is able to produce a better folding, improving the biosynthesis of factor VIII and the secretion. So that's where we are different from every other manufacturer or biopharmaceutical working in this space. So what can we also do for a number of patients that cannot receive this gene therapy because their liver is dividing, the liver cells, the hepatocytes are dividing so fast, like in the pediatric population, in children, where we cannot really treat these patients. Indeed, all the trials that the entire industry is conducting right now is for patients 18 years and older. But as you can see, these patients can be identified in the early years, and especially with genetic screening, prenatal screening. So what we are developing right now is in vivo CRISPR gene editing, which means that in this case, the transgene, the same transgene that we're using, this chimeric transgene, is not only circling the chromosomes, it's integrated into the chromosomes. And we have already published initial work in animal models where we were able to incorporate the corrective factor VIII transgene into the genome. So it's like having the landing in the moon, where we, in this case, are incorporated into the chromosome. That means that even if that cell divides, as happens in children, this cell will maintain the information. And we take the advantage of the albumin promoter. That means that we are not using a promoter that we are incorporating into AV8. So in that case, we have a site-specific integration in the genome. And in that way, we are, as you can imagine, doing something absolutely novel. And this also, we haven't started interacting with the FDA yet, but this requires also a regulatory approach, which is really much more complicated, always focusing on safety. So I'm going to really just keep these slides for you if you want to review them at a later point, because they are very busy, all of them. But just ensuring that you understand that from my perspective, we need to take a look into the study population from obviously the head of clinical development and myself, where we ensure that we have a number of genetic tests that we are performing on these individuals to understand if this is a deficiency or a dysfunction of factor VIII. We also take a look into pre-existing antibodies. We anticipate the risk, potential benefits to subjects. We have all considerations regarding enrollment of children. As I mentioned, in this case would be CRISPR gene editing, or taking into account that here we are not going to be able to enroll healthy volunteers. There are a number of safety considerations very relevant from the regulatory perspective. We need a monitoring plan. We need to ensure that we stagger administration, obviously, in our dose escalation studies. We have study stopping rules, potentially even being able to de-escalate the dose. We look into viral shedding in different fluids, body fluids. Very important to incorporate pharmacovigilance and long-term follow-up. And in terms of efficacy, also absolutely relevant, especially for the pivotal trial. All this is part of the discussion with the FDA. We have a number of aspects that are very meaningful in terms of controlling the disease of the patient. In our cases, what we call annual bleeding rates and annual infusion rate. So we want to see how is the bleeding taking place in these patients, and if they are able to reduce the number of infusions that they receive for factor VIII replacement. That's why it's so important also to take a look into the endpoints. And finally, everything related to dose selection. We need to leverage all the, as I mentioned at the beginning, all the non-human data obtained in animal models and translate that into the dosing that we think is adequate. We still need to have a dose escalation program. And if the transgene expression and consequent treatment effect are decreasing over time, we need to ensure we understand how to do that. We expect that our treatment with our transgene is going to improve both stability and durability of the factor VIII expression in hepatocytes. So with all of that, here is another slide, very busy, where I'm just highlighting the fact that these are single-arm clinical trials. Sometimes we incorporate historical controls. And very important from our perspective is ensuring that we have a number of biomarkers that are going to be incorporated into the study, both in terms of genomics, transcriptomics, epigenomics, proteomics. We also create signatures, and we identify the right patients that are going to be incorporated also from phase 1-2 for our future pivotal trials. And finally, just giving you a sense about the incredible work that goes into an IND filing. In our case, we will achieve the IND clearance for ASC 618, which is this hemophilia A transgene, in July 2021, this year. And it incorporates a very complex type of materials with a module 1 and 2, which are more general, a module 3 on manufacturing, a module 4 with all the non-clinical study reports, and then all details about our clinical study in the module 5. But with all this information, the agency makes a decision and lets you start with the clinical trial. That's what happened to us. And all this information, in our case, was included more than 6,000 pages of documentation. With all of that, I would like to thank you for this opportunity to present our plans and some data. And I don't know if we have some time for a question too.
Excellent, thank you, Oscar, for that really packed presentation. I really love the kind of space analogy. I think it's that whole, probably that cliche thing of like out of this world complexity, literally, I suppose, as a thing. We did have a couple of questions that came in. We do have a few minutes left. If anyone else wants to post a question just in our chat on the right hand side of your screen. So we had one that came in. There's a question: can allogeneic cell therapies be categorized as biologics? This question.
Yeah, allogeneic cell therapy is not what we are talking about today. So we're talking about gene therapies, but yes, the complexity is also very high, and there are several aspects that are also related to gene therapies in the sense that you are manipulating your cells, and you also need a different level of interaction with the regulators, because you are telling them first about the way you are doing this genetic engineering in the cells ex vivo, and then how these cells are allogeneic cells off the shelf are used into patients. So this increases the level of complexity from a regulatory perspective.
Mm-hmm. There's one that's maybe just kind of related to that, and I know I've got some other ones that are more kind of gene therapy related in a second, but it was where stem cells are used in the manufacturing process, but the finished product is clearly not a stem cell. How would the agency classify the finished product? Differentiated cells or another classification?
So again, we're talking about stem cell therapies, and I'm happy to tell you that we have a program on allogeneic cell therapies. By the way, ASC Therapeutics that you see there is applied stem cell, so we're experts in stem cells, and this is an area of extraordinary complexity. And the most important aspect that I would like to highlight here is that stem cells have the capacity for differentiation. And the most important aspect when you're treating a patient is ensuring that these cells are stable and do not differentiate. So we call these cells, especially for allogeneic therapies, stromal cells.
Okay. And there's one other question that's come in, which is an interesting one. So it's like, what do you see as the key bioethical considerations related to CMC aspects with gene and cell therapies?
So bioethics is extremely important with what we do in both gene therapy, but especially in gene editing and CRISPR. We take an incredible amount of care for quality. We have an entire department that really checks independent team all the quality aspects that go into manufacturing. So just using all these aspects, I think, is going to increase our ethical approach to manufacturing.
Fantastic. Well, that is all we've got time for. Oscar, it has come to past the hour, but thank you again for a really great kind of presentation, touched so many different things then, and thank you for responding to those questions as well. Yeah, thanks again, Oscar.